Hello everyone, and welcome to today's live broadcast, Implementing Longitudinal Clinical Whole Genome Research, presented by Benjamin D. Solomon, MD. We're excited to bring you this educational web seminar presented by LabRoots. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Before we begin, I'd like to remind everyone this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, and to get your credits, click on the button located in the bottom left-hand corner of your web page. Also, please notice that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Thank you. Now I'd like to introduce Dr. Benjamin Solomon. Dr. Solomon serves as Chief of the Division of Medical Genomics at the Inova Translational Medicine Institute and is an Associate Professor at the Virginia Commonwealth University School of Medicine. An accomplished scientist and medical geneticist, Dr. Solomon is dual board certified in pediatrics and clinical genetics through the National Human Genome Research Institute. At the Inova Translational Medicine Institute, Dr. Solomon leads the Medical Genomics Division, a group of clinicians and researchers that focuses on providing genetic and genomic medical care, discovering new explanations for genetic disorders, and studying the best ways to integrate cutting-edge genomic resources into clinical practice. Previously, Dr. Solomon held positions at the National Institutes of Health, researching the genetic and genomic causes of both rare and common conditions. The author of more than 100 peer-reviewed articles and book chapters, Dr. Solomon serves as an editor on a number of medical journals, has edited several medical textbooks, and is actively involved in genetic and genomic training and education. Please join me in welcoming Dr. Solomon. I will now turn the presentation over to him.
Great. Thank you so much, Judy, and I'd like to thank the organizers for having me, and to all the folks who are on here. I'm going to go ahead and start on my presentation. I apologize, I think the title is a little bit different than what I submitted, but it comes down to the same thing. And again, I'm excited to talk to you about this, and I'm very happy to answer questions or get into any discussions afterwards. These are my disclosures, though there are no other conflicts of interest. So my talk is going to be loosely organized into 3 overlapping sections. First, I'm going to talk about the background and some of what we do here at our research institute. The meat of the talk, and perhaps the most important thing, will be some of the examples and cases of what we're doing and what we found. And then finally, at the end, if you'll indulge me, I'm going to go into some lessons learned and certainly things that I've come to understand better working in the real world, but also trying to bring cutting-edge genomic technologies to a busy working healthcare system. So first, I'll start with background. So I just want to tell you a little bit about where we are. We are, as the map shows, based in Northern Virginia, northern and a little bit into Western Virginia. We're a decent size, though not a giant healthcare system. We have 6 hospitals, including a women and children's hospital. A brand new one just came up a couple months ago. About 2 million visits a year throughout the hospital system and the ambulatory care network. And we do a fair number of deliveries for a hospital system this size. We have about 20,000 deliveries per year. About half of those are on our main campus that's shown in the bottom slide there, which includes a 1,000-bed hospital, new women and children's hospital. And we have currently actually about 20% higher than that, about maybe 12,000 or so deliveries a year. So we take care of a lot of patients, and we do research on a lot of the folks who end up being participants in our studies. Our research institute was started about 5 or 6 years ago, depending on, again, what you count as the starting point. And we, like many other institutions, have lots of fancy mission statements and ideas about our visions, but it all boils down to trying to do relatively good scale genomic research to learn how to use that information to understand health and disease better, and perhaps just as importantly and certainly much more challengingly, how to put that information back into a busy working healthcare system in a way that, again, helps our patients and isn't just kind of dumping lots and lots of data on patients and the clinicians that take care of them. I do want to mention that right across the street from where I'm sitting now, from the main hospital campus, is this building. This was the former ExxonMobil headquarters. Is now, as of October, the Inova Center for Personalized Health. So we're, as you can imagine, very busy doing lots of retrofitting and building new buildings for this space. It's about a 1.3 million square foot space. It sits on dozens of acres, so there's lots of room for more development, but all dedicated to personalized health and genomic medicine and all the things that many of us like so much. That picture at the top, by the way, that's the governor of Virginia. For those of you who don't recognize him, that was at the announcement about this. And the head that's cut off there, that's the head of our healthcare system, who's a great believer and proponent of personalized or precision or genomic medicine, whatever you want to call it. So the themes of our studies involve trio-based whole genome sequencing. So we do the genomes from mom, dad, and baby, and about 5 to 10% of the time, we also look at genomes from other relatives, aunts, uncles, etc. We generate as much other omics as samples allow at the RNA, epigenetic, proteomic, metabolomic levels, and so on. We try to collect many tissue types and generate data from many tissue types as well. So we collect placenta and blood and urine and saliva and cord blood, etc. When a patient has a condition, such as a baby with a congenital heart anomaly, and they're doing surgery and discarding some of that tissue, we try to collect that tissue both for primary studies as well as validation and functional analyses later on. We collect comprehensive clinical data, so we pull in all the data from our electronic healthcare record system, which happens to be EPIC, and we supplement that with study-specific surveys. And as the diagram shows you, we bring everything together into a unified database to help with statistical power. So whether it's we're looking at a complex disease like preterm birth or an N of 1 Mendelian disease, we have a better background database to help with statistical power. This gives you a snapshot of the number of folks in our 3 main studies. We also have a number of smaller studies. In blue is a study on preterm birth, so it includes both cases and controls, about half cases, half controls. In red is the longitudinal study, which I'll tell you a little bit more about in subsequent slides. And then in green is a study, like a lot of groups have, on undiagnosed conditions. So conditions mostly coming out of our very large NICU. We have about 110-bed NICU of children with conditions that can't be diagnosed quickly by conventional means, so we try to find answers through genomics. So on the left, those are the number of patients that are enrolled. This is about a month out of date, but pretty current. And then on the right is the number of clinical genomes we have on hand, so genomes with matched clinical data that we're using now. And as you can see, there's a lag, and we tend to batch things, especially if folks, which most of our kids are, fortunately, are healthy. We batch and wait and then do them as pretty large units when we sequence. The longitudinal study, which I think it's fair to say in many ways is our flagship study, has a goal of about 5,000 trio-based families. This is a longitudinal study, so we consent them usually about halfway through pregnancy out of 5 OB offices. Those are 3 regular private practice offices, 1 high-risk practice, and then 1 very large clinic for the uninsured or underinsured. We collect the sample types mentioned there, and we generate all the omics that you see there. And this is just a snapshot of the inclusion and exclusion criteria. So we are biased in several ways, admittedly. One is we're biased. We require full trios, so mom, dad, and baby. We're also biased, and this sounds facetious, but it's true, for fertility, right? So these people have to have had a child. And then there's certain criteria you can see there having, for example, to do with cancer or transfusions that we exclude folks because it would affect the omic analyses. I know this slide is hard to see, but this shows you the accrual rate. So we get about 60 to 100 new families a month in the longitudinal study. It ends up being a little more than a third of eligible approach families enroll. And it ends up being about 10% of deliveries in our main hospital. The inflection point that you guys see, if I can use one of these fancy tools here, the inflection point that you see here, that's when we added the what's called the Inova Cares Clinic, which again is a clinic for the uninsured and underinsured. So we started getting more patients when those folks started enrolling. To give you a sense of general demographics in our studies, those three main studies, so the longitudinal study, about a third of people again participate. Our study on premature birth with cases and controls, less than those numbers participated, so about 16%. Those folks were approached after delivery, and so obviously many of those families have other things on their mind when they have an extremely premature child than participating in a genomic research study. And for our congenital disorder study, we have about a 41% participation rate. Again, this is of eligible approached families. This is very general overall demographic data. I'll show you some more specifics in later slides, but it's a pretty socioeconomically and ancestrally diverse group of participants. And though I'm not going to go into this for reasons of time, one of the main reasons that we found that folks don't participate is that the dad doesn't want to get an extra blood stick. So we collect the blood samples from the baby and the mom along with routine clinical care so they don't get an extra stick. But dads, it does require extra phlebotomy. So that's been the main reason in our experience that families don't want to participate. This gives you a slightly better sense of cohort diversity. Again, the map is a little out of date. It's about maybe a year out of date, but it gives you a sense of the proportions. And I apologize because that's hard to see, but each of the numbers here, let me just circle some of these. Each of the numbers shows the number of people that were born in that country. Obviously, everybody born outside of the United States was a parent that came to Virginia to deliver, so we have the most number of folks in Virginia. So these are all where people are born for the genomes. And then on the other side here is just a very simple PCA plot, just showing where people are from who participate. And again, the point is we have a very diverse cohort with people from all over the world. We have a special strength in areas of Central America where we do have a lot of immigrants to this region. I know it goes without saying for this crowd, for this audience, but I'll say it anyway. Having the trio-based data allows us to better consider the inheritance of a variant of interest, and having an ancestrally diverse data lets us consider, we hope it a little more intelligently, the specific allele frequency of that subpopulation. Again, to try to think about, is this a pathogenic or non-pathogenic variant that we're looking at? I think hopefully this will be a point that's very clear, but I always like to emphasize that folks like me, I think of us as the very visible, say, penguins on the tip of the iceberg, interacting with patients, interacting with clinicians, etc. But certainly, and this is a very simplified diagram, but certainly having this amount of data requires a very robust IT network under the surface to store this amount of data, to make sure that we use it appropriately, protect it appropriately, make sure that we're sharing appropriate non-PHI linked data with our collaborators and with certain folks in our group. And it does take a big, very smart, very good team to do this. I think it's something that's underappreciated. And interestingly, of the expenses in our studies, one of the biggest expenses, of course, is generating all the omic data. The other biggest expense is simply storing all this data. And though it's decreasing, it's still considerable, the amount of resources this takes. Then this is just another schematic from some of our ITs of all the different pieces that need to talk to each other and all the different nodes that need to interact appropriately to allow us to do this type of work. And again, most of this, I think, is not appreciated nearly as much as it should be. All right. So with that, I'll thank you for your time and for the first part. I'm going to go on to talk about examples and use cases and some of the things we're finding. Again, I want to mention that I'm just going to be scratching the surface here, and there's many other things that we're doing. I'm happy to answer questions about that, but I just picked out some examples I thought would be interesting. This is, sorry, a little bit cutesy, but this makes the point that I'm going to talk about both starting with simple and then to more complex examples from some of our studies. So to start with, and this might be considered a simple example, though it's clearly not a simple example. This is, I apologize, hard to read. You can refer to the article where we published this, the citations given below. But this is a child who's born, and each of these boxes represents a different test that was done to try to figure out what was going on. So blood tests, urine tests, radiology tests like an MRI, getting a consultation to try to help figure out what was going on. And this is pretty typical in today's world, even with the omic tools that we have, that we do test after test after test, and finally we just throw up our hands and give up and say, okay, let's do an exome or a genome. And I know this is controversial, but one of the things that I would posit is that we should applying these genomic technologies earlier in these children that have concerns for a genetic disease, and we shouldn't wait for it to be a last resort. When we do that, our and certainly a number of other groups, Stephen Kingsmore groups in London, groups in the Netherlands, groups all over, have shown that it's more cost-effective to apply these genomic technologies early. It's certainly better for patient care, better for genetic counseling, better for patients, families, healthcare systems, and society. And in fact, I became interested in this question a couple of years ago when I was still at the NIH about this question of, does it make a difference? And I think there's this idea out there that, fine, we can figure out what's going on and information is good, but it doesn't really change things. And so I started, and I'm not sure why I started, I'm not honestly sure why I kept doing it, but I ended up looking at every single gene involved in Mendelian or a kind of a monogenic disorder. And I only looked at genes where you might find a mutation on germline testing. I only looked at single gene disorders, so not contiguous gene syndromes, as mentioned. I also didn't look at somatic things, such as involved in somatic cancer. But I looked at all of these, and I found that almost exactly half the time you would take care of the patient differently. There'd be a different medical intervention if you knew the molecular cause. And unsurprisingly, in the majority of the time, 95.6% of the time, we would do that intervention early. We would do it during infancy or childhood. I still keep this database updated, and this is housed at the NIH. If you just look up, if you Google NHGRI or NIH and Clinical Genomic Database, you'll get the link up to this. And I spend a couple hours every day updating this with new information. It's a freely available tool, and one can download the spreadsheets and slice and dice it and use it, hopefully, to your heart's content. I think another point, though, and primarily through these omic or genomic technologies, is the point that this field is expanding at what I would call breathtaking speed. So this is a little thing that we publish. It's not on PubMed yet, but it's at the journal's website in case anybody's interested. And in the last about 2.5 years since I published the Clinical Genomic Database in spring of 2013, on average, over 14 new genes were discovered to be involved in Mendelian forms of disease per month. So 14.1 disease, new explanations of disease per month, which I think is pretty staggering. I think the other really interesting thing that came out of our analyses is that despite the fact that exomes and genomes are still pretty new, so the first publications about them, as I'm sure everybody knows, about finding genetic causes were 2009, 2010 through today's methods, and 87% of the new genes were discovered through some kind of genomic, meaning exome or genome technology. I think even more interestingly, as shown here, let me just try to draw this, 47% were discovered with exomes alone, so without doing linkage analysis or homozygosity mapping or candidate gene studies. So just exomes alone in a family or a couple families found the answers, which again I think is pretty impressive. The other point is that if you compare the first half, this is the first half, with the second half of publications, there's even within that 2.5 year or so period, there's a shift where there's more genomics going on, more and more and more, and candidate gene studies are getting used less and less, and it's pretty staggering the pace at which this is changing, which is obviously an exciting thing for scientists, but certainly makes it tough for clinicians and labs and folks to keep up with, to incorporate this new knowledge into medical care. I think the other point that I would make is that not only can we take care of patients better now with this information, in a lot of the cases, this obviously leads to new therapeutic insights. So this is not, this study, which I love and I think is just wonderful work, is not anything that I can pretend to claim credit for. I just heard a talk about this and I've read these articles. I think they're great, but I think it's a really nice example. So as many of you guys would recognize, that child there has a relatively common, by our standards, genetic condition, Muenke Syndrome. And through understanding what was going on biologically by first finding the genes involved and then doing a lot of traditional research, understanding the mechanism by which Muenke Syndrome occurs, the scientists involved in this who are at Hopkins and elsewhere are able to do some work, and that's what these panels are showing, looking at how in a mouse model some of these could be ameliorated, some of these effects could be in fact treated. And I think a lot of this work is very exciting to think about how we could how we could start to manage better, how we could start to treat better these conditions where before there was nothing really we could do other than supportive care. And again, I think the point I would emphasize is that it only comes with understanding what's going on to cause these conditions at the biologic, at the molecular level. So I want to talk about a few examples at this point from our own experience. This slide is just to remind me how to start this story. So I was at a fundraising event, as this slide is supposed to show you, about a year or two ago, and I was with a family who had participated in one of our studies, in a study on preterm birth. And like we usually do, we often invite families to take part in these events. And I talked to the mom and dad for a while. They're really, really nice folks. And then later, when I was doing analyses, found something that I thought was really very interesting and really brought home how much the practice of medicine, the practice of genomics and genetics is changing because of these new technologies. So some of you guys might recognize this man. This is my former boss and still my close friend and collaborator, Max Muenke, who's the chief of the medical genetics branch at the National Human Genome Research Institute. And Dr. Muenke has discovered the cause of a number of conditions. One of the conditions he discovered, as you can see from the citation there, was this condition, which is called, after Dr. Muenke, which is called Muenke Syndrome. And it's one of the most common forms of craniosynostosis. Again, named after him, it has to do with a specific mutation in the gene FGFR3 that causes Muenke Syndrome. And when we did the analyses on our data set, at that point we had a couple thousand genomes we were looking at, maybe 1,000 families or so. We found one family that had the exact mutation that defines Muenke syndrome. So it's a molecularly defined condition. In other words, every single person by definition with Muenke syndrome has this exact mutation. And we found a mutation in 2 members of our cohort, and those 2 members were the child of that family I talked to, as well as one of the 2 parents. And I will not pretend I'm the world's expert in Muenke syndrome, but I worked with Dr. Muenke for years. I've written a number of articles on Muenke syndrome. I've edited a textbook on craniosynostosis, including Muenke syndrome. And in retrospect, I realized that they had subtle signs of this. And looking through the medical records of this child, there were some clues. But again, I think this really drove home the point to me that a lot of our answers in today's day and age is not going to come from the bedside, right? But it's going to come from the bioinformatics suite, and they're going to be the ones, as I'll describe to you in some other examples later, that are going to provide the answers for us. And I think this is changing medicine a lot, and I think sometimes this is a tough thing for physicians and others to think about. Another just example of a case, this was a child also with craniosynostosis, completely separate from the previous case, but this is his 3D reconstruction of his skull showing craniosynostosis. He also had a host of other rather confusing medical conditions. He was small, he was born a little bit early, he had something going on with his kidneys, maybe his adrenal glands, it wasn't clear. Conventional genetic testing couldn't find the answers for him, and a number of tests were done to try to find the answer. And this family took part in our study, and this is a kind of a simplified version of the pedigree. So this is the child who had the condition. Everybody else shown here in the pedigree was completely healthy as far as we could tell, including mom, dad, mom, siblings, and the maternal grandparents. Our conventional pipeline couldn't find the answer, but we found a mutation in the child that all these other folks shown by the stars also had, so a heterozygous mutation. And a typical pipeline missed this, but we instituted an imprinting pipeline and were able to figure out why this kid had this condition rather. And so it's because A, he has the mutation, but B, because he inherited this mutation from the mom. So you only get the condition if you inherit the mutation from the mom, because this is a imprinted gene. And so that was, I think, a nice lesson to us about different ways to use our pipelines to find answers when the initial pipeline isn't able to unearth a cause. But clinically, I think this was even more important. Whoops, let me go back. Was even more important. Operating with 2 different groups. One group at University of British Columbia and one group out in Seattle who have been studying this for a while. We found 4 other families who had mutations in the same gene. So this turned out to be the same, the fifth cause of a condition called Adams-Oliver syndrome. Since then, there's a sixth gene has been found. And again, it really, I think, drove home to me this point. And, you know, we've been involved in other gene discoveries and have more on the slate. But how quickly things move these days and how quickly new discoveries get made, new explanations of genetic conditions are found through these new genomic and omic technologies, and kind of really a breathtaking thing. This is a picture of Jared Roach and Gustavo Guzman, our friends and collaborators at the Institutes for Systems Biology, who collaborated with us on this and get the lion's share for doing a lot of this work and finding this new gene as well as other genes. I've talked about some of these Mendelian kind of N of 1 type cases, and I want to move on now to talk about some of the cases where we combine some of these conditions. So I apologize, this is looks like it's a pretty fuzzy slide, but this is on blood-based newborn screening. So we looked at the genomes of about 1,700 kids that had undergone whole genome sequencing along with both parents. And again, we tried to ask a pretty simple question. You know, if we had whole genome sequencing data, would that make newborn screening more accurate for these kids? And these graphs I know a little hard to read, so let me take you through these. As most of you guys probably know, newborn screening involves several components. One component is called blood-based newborn screening. So they take a drop of blood from an infant's heel, send it off to the state lab, and test for, depending on the state, between usually about around 50 or so conditions. That can be helped by recognizing them early and obviously treating them early. And it's a very good screening success. It's a wonderful public health measure, but certainly like any public health measure, it's not perfect. So what this is showing you on the x-axis here, this is the gestational age. On the y-axis, this is the number of repeat screens. So you can see with earlier gestational age, so these are extreme preemies, you get a lot of false positive newborn screens, right? So you get this kind of boy who cried wolf phenomenon where you don't know whether to believe if they're real or not, and I think sometimes they get ignored. These are the analytes that are some of the most challenging ones that often cause the false positive out of the different conditions that are examined as part of newborn screening. So we looked at 163 genes that correspond to all the conditions that are currently screened for by standard blood-based newborn screening. We looked with, we had these folks were sequenced by two different technologies. In red there is CGI, in blue is Illumina, and you can see on a per gene basis the coverage for most genes is pretty good, but there's certainly genes where the coverage is not good, and as everybody knows, it differs a bit from technology to technology. One of the things that was really interesting, again, is the number of variants and the number of novel variants that we found. So again, on the x-axis, you're looking at all the different genes that we looked at, 163 genes. On the y-axis is the number of predicted protein-altering variants, so missense, nonsense, things that would affect the protein sequence. In purple are the number of ones that we observed in these about 1,700 babies, the number of protein-altering variants, again, on a per-gene basis. And in brown are the ones that have been deposited at that time. We published this about a year ago, but in publicly available databases. So the lesson from this, or at least one of the things that we learned, and I think this is known, but it's nice to quantify some of these things, is that if we sequence any population, even a pretty small one, we're only looking at 1,700 patients here, you get a lot of novel findings, a lot of things that could be deleterious, might be deleterious, could in the context perhaps of a recessive disease be pathogenic, and so on. I think and so I think we as a community, as a research and clinical community, have a lot of responsibility to help contribute to these databases so that more things are represented, and so that we again can consider the pathogenicity, the deleteriousness of variants, hopefully a little better. And certainly there's a lot of room for these to grow, and I know there's lots of wonderful efforts like what ClinVar is doing to allow these to be even better. This is a table that I know is a little hard to read, so let me walk you through it. This is basically the head-to-head comparison of newborn screening. So let me just use the drawing tool. So standard newborn screening versus genomic sequencing shown here. And so what you can see is that we're we're able to resolve some of the false positives in newborn screening. So we're able to resolve some of those with genomic sequencing. But a lot of the trade-off is you get a lot of uncertain events or uncertain results from genome sequencing. And again, there's a lot that as a community we can do to help with that problem, such as depositing things to public databases and so on. One thing I'd explain is that this is a big number here, 45,000. But we talked about 45,000 different tests, right? So even though newborn screening is kind of one screen, it's a combination of looking for lots of different conditions. So each considered each of those a different test. And again, I know this is a little confusing, but I invite people or encourage people to go and look at this paper. And I think, you know, it was very illuminating for us about what's helpful from genomic sequencing and what parts are unhelpful. And certainly certain genes work really well to look at, and it's very helpful, and other genes aren't helpful at all. The most common cause of congenital adrenal hyperplasia due to a pseudogene isn't something that with whole genome we can look at well. And other conditions we can't look at well either, like congenital hypothyroidism, both because those genes are very polymorphic and because there's lots of non-genetic causes of congenital hypothyroidism. One of the things, though, that's talked about a lot is this idea that we can augment newborn screening. We can add things to newborn screening that aren't currently included, but that by the standards by which we include things would be included. We can detect them, there's a treatment, etc. This is a child with, as you can see, pretty massive hepatosplenomegaly, so a really big liver and spleen. It was thought this was because of some kind of anatomic shunt, some kind of vascular lesion causing blood and so on to build up in those organs. We did whole genome sequencing. This was a child in our studies, and we found that the child had biallelic, so recessive mutations that cause this child to have Muenke Syndrome. And I remember very vividly, I was at a conference in California, and Dale Bodian, one of our lead bioinformaticists and the person who, again, gets lots of credit for solving a lot of these cases, called me and said, you know, could this patient have Muenke Syndrome? And I picked up my phone, and I have a link on my phone so I can go into the electronic healthcare record, and yeah, it made sense. And so we were able to find this answer faster than it was figured out clinically, and we were able to get the kid in within a day or two to a biochemical geneticist in the area, Brendan Lanfer, who's now at Mayo, who's a wonderful geneticist, and then talk to Denny Porter, who's the clinical director at NICHD and one of the world's experts in this, to talk to the transplant team to better take care of this kid. So to give IV a medication called IV cyclodextrin to hopefully have his liver hold out till transplant, and he made it, and he's now status post-transplant. But to talk as well about intrathecal cyclodextrin for neuroprotection. The other thing that's not as rare a condition, but we found about 15 kids with this in our cohort of 1,700, is G6PD deficiency. Many of you guys know that G6PD deficiency is the most common enzymatic deficiency in humans. We found about 15 cases of this in our cohort. It's an X-linked condition, and kids can look perfectly healthy until they're exposed to certain medications, certain antibiotics, certain antimalarials, certain types of food. The most famous one is fava beans. And then they get these hemolytic crises, as the cartoon is showing, and it can be very severe. They can need transfusions. They can it can be lethal in some cases. And knowing about this is a simple thing that you just hopefully try to avoid some of these stressors. It can make a big difference. Again, G6PD deficiency is not something that's on newborn screening in the Commonwealth of Virginia. It is just north of here in DC, as in other states as well. But again, I think it provides fodder for this thought experiment about how we can use genomics to better take care of patients and to pick up conditions, hopefully, a little bit earlier. We're also doing similar studies now about hearing loss and immunodeficiencies, and should have similar data coming out, hopefully in the next year or two, on those types of conditions. I want to give one example now, and I promise I'm getting close to the end, of how we look at complex disease modeling. So previously I talked about Mendelian conditions, monogenic conditions, either alone or in combination with others, like in our newborn screening study. We also look at multifactorial conditions, such as preterm birth. And so trying to use all our clinical and genomic and multi-omic biologic data to try to understand what's causing preterm birth. And this is in, I should give lots of credit to, again, the folks from Institutes for Systems Biology, Ilya Shmulevich, Gustavo Guzman, Jared Roach, Brady Bernard, and others, as well as the folks from GNS Healthcare in Cambridge, who have been very terrific with a lot of the modeling. So this figure is just showing you that if we have just the clinical data, so if we take all the clinical data we can and scrub it to pieces and make it and arrange it as nicely as possible. We max out at an area under the curve at about 65-ish%, meaning we can't predict whether or not someone's going to have preterm birth much better than that, and it doesn't get any better, whether we have 1,000 or 10,000 patients, right? Which is better than flipping a coin, but not much better. And obviously, it'd be great if we could understand the causes of preterm birth, because then we could predict who might have it better and better take care of those folks prenatally. And obviously, just like the Muenke Syndrome, hopefully that would lead some windows into therapeutic research. When we do a traditional GWAS-style approach, again, we're statistically underpowered, and unsurprisingly, we don't find answers there. We get a few blips that look like they have some significance, but nothing that really jumps out, really would pass muster here. And again, this is very expected. Things get interesting though when we combine some of these data types. So here we're combining with a random forest analysis. We're combining all the genomic, the whole genome data with the clinical data, and we see some separation. So in red here, let me just draw a little bit. So these red dots, the more red would be the earliest preterm, and then blue dots are term, and you can see that there's a separation, right? So a perfect separation. All the red would be huddling in one corner, and all the blue would be very separate. But we're starting to see better separation now, better predictability, in other words, when we're combining clinical and genomic data. This is a bubble plot, and I know this isn't informative by itself, but things get even more interesting when we combine multi-omic data. So now we're looking at DNA-based data from the whole genome, we're looking at clinical data, we're looking at RNA data, we're looking at epigenetic data, microRNA data. And so the bigger the circle, the more statistically significant the variable. And so we're looking at, in blue, the most significant variables, and it turns out that some of those are clinical, I think one DNA, some RNA and epigenetic, and the point is now we're getting to the amount where we have some statistically significant biomarkers that we're now validating, but those are the kind of things that you could use in real time, right? You could pull off onto a chip and quickly test to see if a woman who's pregnant, or even maybe before she's pregnant, is at risk of preterm birth. Unsurprisingly, and kind of happily, when we do network analyses, the networks that are implicated are a lot of the ones that make sense in terms of causes of preterm birth. Things like immunologic networks and networks defending against bacterial infection, collagen networks, and so on. And again, we're still working hard. This is preliminary data. We're also, again, with our collaborators at the Institutes for System Biology doing traditional proteomic work. So as I mentioned, we have a good placental biobank. So we've looked at biomarkers, proteomic biomarkers in placenta to help us understand what's different about a preterm versus a term placenta. And now we're validating those in the blood samples of women from blood samples that are collected at about 20 weeks gestation when they do the oral glucose tolerance test to screen for diabetes mellitus. Again, looking for biomarkers that can help explain preterm birth, which has a big impact on society and certainly on the individual patient level. So if there's anything we can do to understand it better, prevent it, it would be a big public health win. I want to mention, this is the last slide before I go into my final section, that we have some neat findings that are perhaps not quite as immediately clinically applicable, but I think very exciting scientifically with de novo variants. And so we have a Nature Communications article that came out recently, and then it says under review, but we just have the second paper accepted by Nature Genetics, and it's showing some unique correlates of de novo mutations, and including with maternal age, and now we're looking at other factors such as assisted reproductive technology and other things that help us understand why de novo mutations occur, driving both disease and evolution and so on, and I think just as importantly, architecturally, where in the genome they occur. And again, the lion's share of the credit of this goes to Wendy Wong, one of the bioinformaticists here, and she's worked very hard with Dr. Jacobson there, Christian Gillison, and folks at Radboud University in the Netherlands on this, which I think is some of the, not immediately as translatable work, but certainly some incredibly scientifically exciting work. So at the very end, I want to talk just very quickly before I take questions about some lessons learned here. First of all, and I hopefully this is something that makes a lot of sense, the diagnostic paradigm is shifting. So this is, as many of you guys will recognize, the show House. And previously, as recently as a decade ago, the model where we tried to diagnose someone would walk in the hospital or get seen at an outpatient clinic. We do one test, that would be wrong or wouldn't give us an answer. We do another test, that wouldn't give us an answer. Finally, some really bright Dr. House would figure it out, or nowadays we do genomes, we figure out the answer. And things are shifting from the doctors being the one deciding what the test is to the doctors being the one helping interpret and help the patients understand and manage the conditions with that data. And I think that type of change in medicine is certainly not an easy thing, and I think that's a hard thing for many clinicians to take, that they're not the ultimate arbiter of a lot of these things. But certainly I see things in my field and in many fields that involve genetics, and many fields of medicine do, changing a lot. The second thing along those lines is that I think it's very important to manage perceptions. And I don't know where the audience thinks we are on this graph of hype or of expectations. But one of the things that I think is certainly true is that we are at the Wright brothers stage, or the Model T stage, as my boss likes to say. So we're very early at the beginning of being able to understand what a single genetic variant, how it affects someone's health, let alone in concert with lots of other genetic variants and with all these other non-genetic factors. So we have tremendous amount of work to do to really be able to interpret a genome, let alone a genome with all the other omics and all the other clinical data. So I think the glass half full is that we have in some ways job security. The glass half empty is that I worry about sometimes about all the buzz and the hype being counterproductive to this idea of precision medicine or personalized medicine. Along those lines is that I think it's really critical to provide help and guidance, right? As the picture shows you, this is from the New York Times article on an unrelated subject. But doctors, as we know in this country and elsewhere, are very busy and are expected to see a lot of patients and manage a lot of complexity. And adding much more complexity to the system isn't necessarily going to be helpful, right? So I think it's critical that we do a lot of, and this is going to sound I do not mean this to sound disparaging, but we need to do a lot of hand-holding and a lot of helping for these clinicians to help them interpret and use these data, because certainly genomics and precision medicine is not something that's taught well now in medical school or medical education, and definitely 10, 20 years ago wasn't even thought of. So things are changing, and we need to do a lot of helping for the folks out there. Despite the fact that the last couple slides may sound a little negative, I did not mean it to be, but I really do think it's a wonderful time to be a geneticist and genomicist. We have enormous challenges, but it's an exciting time, and I think the kind of hypotheses that we can address, the questions we can ask, is just wonderful these days, and I think there's tremendous promise to come, but it's going to take a very, very lot of hard work from a lot of very hardworking and smart people. With that, I'd like to thank you for your attention. Obviously, too many names to list here, especially my boss, the former head of the NCI, John Niederhuber, has been a wonderful mentor and leader of our group. All the folks that work very hard at ITMI with me, and all the other.
Sorry that we accidentally cut you off there. Thank you, Dr. Solomon, for that informative presentation. It is time for Q&A, so if you have a question you'd like to ask Dr. Solomon, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. So our first question is, what did you mean by different pipeline to use in the analysis when you talked about imprinting genes and craniosynostosis?
Different ways. Sorry, there's a lot of different ways I could answer that. I think the fundamental point, though, is that there's not one perfect analysis tool, or as we call it, pipeline, that will solve every single case, even if the answer is there. And when we approach these cases, we use a combination of different pipelines. So we have our own in-house pipeline that primarily Dale Bodian, one of our senior bioinformaticists, developed. We've collaborated with great groups that I like very much, from Personalis, Ingenuity, the guys from ISB, their pipelines, and there's no one perfect pipeline, and certain pipelines or analysis tools will provide answers and others won't. And I think the important thing is that we and others are constantly iteratively improving and adding different pieces to our pipelines to try to get them to work better, to find answers. And every time we do that, there's always a new case where we find an answer by looking through a different facet. That imprinting piece was an example of a little piece to add to a pipeline and kind of a filter to detect changes that would affect imprinting. But I think that's where a lot of the fun and excitement lies, is that if I do a genomic analysis of a patient, I don't find an answer, it doesn't mean the answer is there. It means that we need to look harder, look in a different way, and that maybe with new tools, new pipelines, new knowledge, we can find the answer. I think that makes it certainly challenging, but also very exciting and rewarding.
How can we overcome the coverage concerns of using WES or WGS as a tool of diagnostics in the clinic?
Yeah, that's a great question. It's a very hard question. So one thing that's really interesting, and I'll quote some folks I like a lot, like Les Biesecker from NHGRI. Traditionally what a lot of labs do, whether it's on the research or the clinical side, is we'll find something through next generation sequencing, either, as Judy mentioned, whole exome or whole genome sequencing, and we'll validate it by something like Sanger sequencing, right, to make sure that the variant of interest is really there. And statistically, what's shown, right, is that a lot of the times the next generation data is more believable than the Sanger data. So if they don't agree, it's not necessarily that it's an artifact that's not in the next generation data. It might be that the Sanger data is wrong. And so there's a lot of groups that are looking at this to try to provide more statistics. I think one of the groups that's doing this really well is the lab Invitae, and groups like that, to try to show that statistically having certain coverage up to a certain level provides adequate data to results, to provide to clinical results. I think one answer is that things are getting cheaper. We can do more and more high-depth coverage and so on, and we can believe things more and more. But on the flip side, there's a lot of statistics that can be done to show how believable things are, and I think a lot of that work is coming out. Certainly, a lot more work needs to be done. But again, I think this is the bottom line is that the field is changing very quickly, and because it's changing so quickly, a lot of the questions about things like what's the optimal coverage, or do I need to validate this by Sanger? I think those are still being answered, and it's tricky because the field is moving so quickly and being pushed forward so quickly.
Could you clarify the clinical utility of variants found in complex disorders or hits found in GWAS?
Yeah, I think it's a very good question. It's again, I think it's a very difficult question. In many ways, precision medicine represents a couple things, and I think and it represents different things to different people, I should also say. If you read Francis Collins and Harold Varmus's article about, I believe about a year ago in New England Journal, they talk about Obama's precision medicine initiative, and they focus on a couple different things, right? So they're focused on cancer genetics or genomics, right? Somatic cancer profiling, and they focus on pharmacogenomics, right? And so there's certain things that I think are ready for prime time in precision medicine implementation, right? And those are two examples of them. I gave a lot of examples also of rare disease diagnostics and how we can solve or end diagnostic odysseys and provide more answers to folks. I think a lot of precision medicine requires a lot more work and much bigger data sets till we can extract meaningful answers out of them. And I think there's a lot of initiatives to do that. But in other words, we need much larger and much more rigidly constructed databases, not rigidly constructed, robustly constructed databases to answer some of the causes about complex conditions. And these are a lot of the conditions that GWAS studies looked at, obesity, asthma, autism, developmental delay, height, all these things that are these complex multifactorial conditions. And so initiatives like the Obama's Precision Medicine Initiative, the VA, the Million Veterans Initiative, the UK 100,000 Genomes Initiative, a lot of the genomes, the hundreds of thousands of genomes that they're doing in other countries like China and so on. It's only with data sets that size that we're going to be able to have an answer for an individual person, right? I think a lot of the challenge now is we can look for markers, right? We can look for the susceptibility factors, and they may be meaningful on a population level about the risk of a condition, but on an individual level, you know, does this person have a higher or lower risk of, say, preterm birth or asthma or diabetes? They can't really be applied right at the bedside yet. So I think certainly lots of people recognize this, but a lot of more work needs to go in through these initiatives to provide answers that will be meaningful at the bedside.
I would like to once again thank Dr. Solomon for his presentation. Do you have any final comments today?
I'd simply like to thank everybody. I'm happy to talk offline to folks who have questions about collaborations, questions about our research. I wish everybody lots of luck, and I hope that we all working together can find a lot more answers for our patients and better take care of folks in our societies through some of these precision medicine tools. Thanks so much, guys.
Another question has come in. Hoping you have just a quick moment to answer it. Using available tools now, which is better to use, target sequencing or WGS or WES for diagnosis?
That's a really good question. It's a very hard question. I think there's not a single answer, right? Because it's very context dependent. Certain patients, we can do targeted tests and find an answer, and it might be a specific targeted test, right? Just look at one gene or look for one mutation. But in other cases, we can do kind of larger, but still considered targeted tests. So we have a big group of cancer genetic counselors. They're doing what are called targeted tests, but they're not traditionally what we've thought of as targeted tests. They might do a panel for dozens of cancer genes and someone with suspected familial predisposition to cancer, right? And so things are being rapidly changed. What's very clear is that through these genomic technologies is that there are many cases that are atypical these days, right? That patients have a mutation in a gene that don't really look classically like other patients who were previously described. And that makes perfect sense, right? So, 10 years ago, if we wanted to figure out the cause of a gene, we'd get 100 patients that looked exactly alike, and we'd sequence them, or maybe for a candidate gene, we'd find the answer, and by definition, all those patients would have the same condition, right? Because you a priori defined them as such. And so you'd only look at that gene in the next person if that person looked exactly like the original person, right? Or a lot like that original person. And things are being changed dramatically, so we're seeing all sorts of atypical cases. We're rewriting the textbooks about penetrance and expressivity, and things are changing a lot. What I have certainly found myself as a practicing pediatric geneticist, and I work very hard, I don't pretend to be the very best geneticist, but that more and more we're finding answers in genes that we wouldn't have suspected to be involved for a certain patient in an atypical case. And so more and more we're going towards an exome or a genome early. Again, I know that's a controversial thing, but I think it's that really is the best way to provide answers in many of these cases. However, I think one of the most important points about that is that part of what makes sense for a patient, and what I think one of the lessons of precision medicine, is that we want to do what's best for the patient, right? So some families, some patients might want to do genomic or exomic or whatever sequencing early, and they're fully informed about the secondary type data that might be found, and so on, and the fact that there's not complete coverage, and the risks and the benefits. And some patients want to do that, right? We encounter those patients all the time. We also all the time encounter patients that say, no, I want you to do a very targeted test. I don't want this full omics. I don't want to even have the chance of finding out other stuff, even if that stuff's supposedly going to be masked. That's just not right for me. And I think one of the most important things in this is doing what's right for the patient. And when you take two patients or two families with a very similar conditions, one family might feel right for them to do genomics, do genome or exome sequencing early, and for another family that's not what they desire. And I think one of the most important things is that we inform patients and families and research participants of the risks and the benefits, and that we honor what really feels right to them. I think it's a really good question, and I think there's a lot of work that needs to be done to better show how these techniques are effective, in which situations they're effective, and in which situations, frankly, they aren't helpful. Yeah, great question. Thank you.
Thanks again, Dr. Solomon. This is an educational webinar and thus offers free continuing education credits. So after the webinar is over and to get to your credits, click on the button located in the bottom left-hand corner of your web page. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through November 11th, 2016. You'll receive an email from LabRoots letting you know when this webcast will be available for a replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.